Well, first of all, thank you organizers for giving us a chance to present our current work. And second, of course, I didn't know that I'll set a pretty challenging task to draw the audience back from the world of computers and video games back to our everyday science. But we'll see how it works. So it's again, the work is along the sequence analysis and applying modern protein language models for predictions. In our case, we're trying to predict B-cell epitopes. So before we get to the kind of mathematical things and machine learning, I just want to introduce the audience to the biological problem, what this all is about, in case you haven't been involved in any immunological projects. It's part of the immunity, and there are different types of immunities in higher organisms. Here I kind of borrowed the image from Sigmund Aldrich that outlines two types of immunity. One is cell-mediated, and so kind of starting from the top, we all, like humans and many other higher organisms, face every day with millions of different organisms, and not all of them are friendly. So there are pathogens browsing around and trying to kind of sneak into our system. And it can be bacteria, it can be viruses, and so on. And cell-mediated immunity is actually started from macrophages and dendritic cells that are trying to engulf those pathogens and degrade them inside the cells. And once they degraded their proteins, they present them on the surface and trying to present them to T cells that are floating around. And once the T cell determines that this is a fragment coming from the foreign protein, that it's not a host protein or not part of host protein, it immediately sets the alarm and becomes activated, generates tons of cytotoxic T cells that start looking for infected cells and kill them. And on the other side, it keeps some set of these T cells in memory, kind of they call memory cells. So in future, whenever that pathogen appears again, the immune system will quickly react. And another branch of immunity is called humoral immunity, when the B cells deal with whole pathogen. So it's not degraded somehow, but it deals with proteins on the surface of that protein or the pathogen. And B cells have these BCRs, so it's B cell receptors. And once they bind to that foreign protein, they initiate again activation and generate tons of antibodies that attack free available pathogens. And so our project is really about this branch of immunity. So we want to predict, based on the protein sequence, what would be potential immunogenic part of within that sequence, what would be the actual piece of that protein that would bind to B cells, or rather B cells bind to that, and kind of it determines the B cell epitopes. So there are two types of B cell epitopes. One is conformational or discontinuous epitope. It's on the left. So you can see that antibody with heavy and light chain bind to their target, and the residues that they actually bound to, highlighted in red here, they may be in the sequence far apart. So as far as, for example, in this case, as 100 positions apart. Another type of epitopes is linear epitopes. It's fairly short part of the protein that kind of recognized by the antibody. And so why we would like to know, and why we would kind of get into that field and predicting B cell epitopes, there are three major fields. It's designing tools or diagnostic methods that would allow us to diagnose, for example, whether a person has been already infected with a given pathogen. And I guess recent story, you know, about this COVID-19 epidemic. So there are tests to see it's antibody based. So it's actually shows whether your blood or circulating fluid already has antibodies against the virus. There is another direction is developing epitope-based vaccines, and the last one is designing antibody-based biologics that actually attacked either pathogens or cancerous cells. And when you look at this, so the practical consideration is actually develop tools for this is linear epitopes. They are smaller, they're easier to synthesize, and easier to scale up for the large scale production. So that's why we focused our prediction method on linear small epitopes. Previous methods were trying to kind of look into what may bring the propensity in the protein sequence, like what exactly drives the recognition by B-cell antibodies, B-cell receptors and antibodies to bind those things, and normal things like physicochemical properties, consider it hydrophilicity, polarity, how exposed that residue, and so on, and trying to go along some sliding window, trying to see if there is any profile that would give a propensity score for antigenicity. And some simple machine learning approaches were to combine a few of these physicochemical properties. So now, the field actually was stalling, meaning that prediction is still pretty random for this B-cell epitope. So lots of things to explore and improve. The current situation is that with deep learning, we have some additional input space that we can either use 3D structures predicted by AlphaFold or imply protein language models that I'm going to describe quickly in the following slides. The reason for protein language models kind of promises that it seems to be from other unrelated projects. It seems to be those embeddings that these models generate. They actually bear some signal for solvent accessibility, for distant relationship and other things. So it looks very promising. So what those protein language models, again, I'm sure most of you already got familiar with that, but just to put everyone on the same page. So it's essentially a large language model, which comes from NLP, from natural language processing. It involves 3 steps. So we have a sequence of any length of any amino acid composition. We tokenize it, and then we run large step. It's a pre-training of the language model that takes millions of different proteins and tries to, so we have, again, different position masked out, or so we force the model to predict what those masked positions are, what amino acids supposed to be at those positions, or we also force what would be, given the sequence, what would be the following symbol or the following amino acid in the sequence. And the last step is having that model pre-trained that generates those embeddings. Can we also apply those embeddings to different machine learning problems, which this step is called fine-tuning. And I highlighted it red, so it means that kind of to indicate where our project is. So we're using someone's already pre-built machine learning models, protein language models, and we're trying to fine-tune it for B-cell epitope prediction. So at the time we started this project, we found a few already published protein language models that already have high citation rates. So we explored them and tried to apply to different problems and see how they perform. And out of all these available models, we've, by different criteria, we picked protein T5. This is a transformer model developed by Burkhard Ross group. All right, so how we get data? Always we need some data for fine-tuning, which is supposed to have some labeling, so for train the classification model. So two major resources we used is immune epitope database, ADB, and so and the second one is bacterial and viral bioinformatics resource center. Quickly, it's all kind of selection criteria that sequences within set and between the sets supposed to be non-redundant. We pick only those proteins that come in from the pathogens and considered only limited linear epitopes. Once we split the ADB data set, which was over 900 proteins, we split it on the training validation subsets and put aside control set that never was seen by the neural network. So we ended up with control set of 13 proteins and blind set of 175 proteins, and the number of amino acids in them reflected. So we built two alternative models just to see how it would perform. So one is 1D convolutional model neural network and another bidirectional LSTM. And we compared it with two methods supposedly prominent in the field. The reason I'm saying is that, for example, IEDB database as a default method for prediction of B-cell epitopes provides BPPRED version 2. BPPRED 3 just recently got published, so it's kind of gaining popularity. And you can look while I'm talking on the column, so there are different metrics for performance measure. I prefer Matthews correlation coefficient, which is the last one. It's easy to interpret, so the closer to 0, it means random prediction, while higher, closer to the 1, it's a perfect prediction. So you can see our method performs the best for the control set, and BPPRED2 actually pretty low in performance, meaning it makes a random prediction. BPPRED3, while it also uses protein language model, it uses ESM protein language model, so it seems to be underperforming. While with the blind set, we do observe some drop in the performance, so we are curious what is happening, and did some small analysis of the data sets. So if we generate a t-SNE plot on the embeddings for the all proteins used in this project, we can see that validation and training set covers pretty much all space. It's gray dots around. Whereas control set and blind set, they kind of slightly separated. So that was kind of curious, something's going on, even though they were completely randomly drawn from the database and used non-redundant. So we looked further. One major difference that we have a slightly different phylogenetic distribution. So in the control set, we also have fungi and other eukaryotic parasites, whereas in blind set, only viruses and bacteria. And the length distribution of the proteins also seem to be shifted. So in the blind set, they tend to be shorter proteins. So that may contribute to this difference in the performance. So on that, I will conclude. So most of the work is done by my student. He runs that in the GitHub, and the models you can find on this Hugging Face. So thank you.
Thank you. We'll take time for one question, if there are any. Yes, please.
Do you think the difference between your control and line says is because when you have the composition of these eukaryotic or fungal?
Yeah, so from our different exercises with those protein language models, the embeddings generated by different protein language models to viruses, bacteria, and eukaryotic organisms, they seem to be different, and it's most likely attributed to the different amino acid use and different overall composition. So
it's not something that you can easily identify, like this has the feature of fungal epitope versus
we're planning to explore that, because it certainly there is some bias, even though the training set itself has everything, but the control and blind set somehow differently stay apart.